So we've been talking about mutations in DNA, and I'm going to discuss today a specific disease that's caused by a single mutation in DNA. This disease is sickle cell. Sickle cell is a serious disease which can be deadly if not treated, and even if treated, it can be deadly, and it's caused by a single nucleotide mutation that causes an amino acid change at a crucial part in the protein. On the top, we see the normal, healthy DNA sequence, and on the bottom here, we see the mutant version of the DNA sequence for this gene, and we see that there is an A that is changed to a T. So this single nucleotide change causes a changed amino acid to be called for during translation, and the protein functions very differently because of that single amino acid that has changed. Now we could see here that there are some red blood cells that look pretty normal. They're kind of a donut shape, and these do not have the mutation, or rather, they're not affected by the mutation right now. We'll talk about why in a couple of minutes. But this sickle cell shaped blood cell normally would be shaped like these other ones, but it has been deformed and is no longer functional as a blood cell because of this mutation. Now, how does this one mutation in a protein affect the entire cell? Well, it's because of the behavior of this protein when it's mutated. Sickle cell occurs because of the mutation, and what happens is that this protein changes the shape of the cell. The changed cell shape to look like a crescent moon or a sickle causes the cells not to work correctly. They're no longer able to carry oxygen and deliver it to the tissues of the body. And these shapes can also get stuck in the transport vessels of your body, and it could cause an occlusion, which is just basically a buildup where normal blood cells cannot get through to deliver oxygen to the body or to those specific tissues. So symptoms include widespread pain in the body due to tissue hypoxia, so tissues not getting enough oxygen, insufficient blood supply to tissues in addition to or contributing to that hypoxia, and then, of course, damage to the tissues because they're not getting enough oxygen and the wastes are not being removed from the tissues either. Other things that might need to be carried in the blood. So normally, a red blood cell is just packed with hemoglobin proteins. You've probably heard of hemoglobin before, and this image depicts a red blood cell. It does not have a nucleus, so red blood cells are one of the very few cells in the human body that does not have a nucleus, and it is just completely full of hemoglobin proteins. So red blood cells can have this very specialized kind of lifestyle because they're only active for a relatively short amount of time. I believe it's under a month. It's like 20-something days. And so they're basically synthesized with all the proteins that they need. They can't make more proteins because they don't have their own DNA. And so they start off with all the hemoglobin protein they're going to get, and that's how much they have until they're retired and recycled. So hemoglobin and red blood cells are supposed to absorb oxygen from your lungs and then travel through various vessels of your body and go to various tissues and deliver that oxygen to the tissues. The oxygen itself is actually bound to the hemoglobin molecule. So those hemoglobin proteins are going to bind to oxygen at the lungs, and then they're supposed to release oxygen to tissues that are deficient in oxygen. So this should happen constantly in your body. I also wanted to note that this image, even though it's showing that there are lots of red blood cells in the hemoglobin, or even though this image is showing that there are a lot of hemoglobin molecules in the red blood cell, not the other way around. Hemoglobin molecules, one, are much smaller than these dots that we see here, and there are many more hemoglobin molecules in a single red blood cell than what is shown here. So there are, I think, over, I think it's 1.5 million hemoglobin molecules per single red blood cell. So they're literally packed full of hemoglobin molecules, because that is the red blood cell's only function. Our main function is to carry the hemoglobin and have it pick up oxygen, release oxygen, and repeat. So the hemoglobin molecule is actually made up of 4 different subunits. So each of these subunits is a different peptide chain, and each of them is very similar. So there are actually 2 copies of 2 different genes that are shown here. It doesn't really indicate it too well with the colors, but you'll see that they're labeled alpha and alpha here, and then beta here and beta here. So we have 2 beta chains and 2 alpha chains, but the alpha and the beta are not that different. And in total, we have 4 different subunits. Now they are always bound together. And what do we call this level of protein structure? Anybody remember? It's a quaternary structure. So this protein has a quaternary structure that consists of 4 different subunits. And each of the subunits, as I said, is bound to one another. So we might want to view the entire hemoglobin molecule as a single kind of spherical or globular protein, and that is shown here. It's not showing it exactly the same as we saw it in this image. It's showing the 2 beta subunits on one side and the 2 alpha subunits on the other side. Either way you look at it, doesn't matter. I also want to point out here that there are 2 different conformations of the normal hemoglobin protein, or hemoglobin molecule, with the 4 subunits. That is the bound version and the not bound version. So this is the bound or relaxed version, where it's bound to oxygen, and then the unbound version, which is not bound to oxygen. So let me just show you a short video of this hemoglobin going between its 2 conformations. It's a subtle shape change, very subtle conformational change compared to some things that we've seen in the past. And this is the deoxygenated version, and then this is the oxygenated version. So again, really subtle changes, but there is a shape change depending on whether or not this molecule is bound to oxygen. Now we start to get a little bit more complicated, because each of these individual subunits can bind to a single oxygen molecule or a single O2 molecule. When one of these subunits goes from the bound to unbound, or reverse, unbound to a bound conformation, then it affects the conformation of the other subunits and makes them more likely to go to the same conformation. In addition, you see that when the beta subunit 1 is not bound, it is in the tense conformation, so it's not bound to oxygen, it has this little pocket, it has this little indentation, and this is actually a hydrophobic indentation. What do we mean by that? Well, simply all we mean is that this indentation has some amino acid that is hydrophobic, and so not necessarily very common. Normally we talk about the surface of a protein being hydrophilic, but when the hemoglobin molecule is not bound to oxygen, we do get that little surface feature, and this is going to come into play in a moment. So that was for the normal version or the wild type version of the protein. Here we're looking at the mutant protein. The mutant protein has this unique feature here. We see that there is a little piece that sticks out. And interestingly, this piece that sticks out is also hydrophobic. So it's a hydrophobic little bump that we see here. And that is present whether the subunits are bound to oxygen or not. But I think you might be able to predict where this is going, because once we have a molecule that is in the tense state and not bound to oxygen, we have a hydrophobic bump, which sticks out, and a hydrophobic pocket, which is an indentation. And that would not be a problem if we just had a single hemoglobin molecule by itself in a cell, but we don't. We have that 1.5 million hemoglobin molecules in a single cell. So what's going to happen is when many of these hemoglobin molecules in a single cell are mutated and have this hydrophobic bump or protrusion, then you're going to get the aggregation or the clumping of hemoglobin molecules. So they're going to fit together like this, in the corner here, so that just notice that this hemoglobin molecule is turned 180 degrees clockwise, and this one is flipped vertically, and we start to get this aggregation again. So this is the hydrophobic protrusion, and it associates with the hydrophobic pocket of another hemoglobin molecule. Again, because what's the rule for hydrophobic interactions? They want to reduce interactions with hydrophilic materials. Inside of the cell is filled with water, which means that it's hydrophilic, or it's hydro. And so that means that these features, both the protrusion and the invagination, are going to associate better with each other than with water, and you start to get these clumps of hemoglobin. Not only are hemoglobin molecules all clumping together, but they actually form these really long fibers. And so you get these linear structures that look like this. We started to see that on the previous slide. And these fibers are actually so long that they can distort, when they do distort, the red blood cell. And so we see that here. It goes into the sickle shape. So again, this sickle shape is due to hemoglobin proteins clumping together and making these long fibers, lots of long fibers that stretch out the cell. Now, in addition, this cell is not functioning as it should be, because once these hemoglobin molecules are in this fiber, they can no longer change back to the shape where they could bind oxygen. So they're staying permanently in the unbound state, the tense state. They're never going to go back to this relaxed state where they could bind to oxygen. So unfortunately, this can occur, or the polymerization, formation of fibrils, can occur exactly when an individual needs oxygen. Now, most of the time, this mutation occurs as in individuals that are heterozygous, which means that they have a copy of the mutated gene and they have a copy of the non-mutated gene. And that's why we can see red blood cells in a single individual that are intact and functional, in addition to sickle-shaped blood cells. this, again, is just this single change in a single nucleotide that codes for a new amino acid. So the original amino acid coded for was not hydrophobic. The new amino acid that's coded for is hydrophobic, and it causes this protrusion from the overall hemoglobin molecule, which in turn is then going to cause formation, aggregation, formation of a fibril, and it's going to lock the hemoglobin in the unbound conformation, so it's not going to bind oxygen anymore, and in addition, it's going to completely distort the red blood cell. And so that completes our lecture on hemoglobin and the mutation that causes the sickle cell condition.